You've joined us for UltraLife Today. I'm Josh Bell, your Chief Product Evangelist with UltraBotanica. I'm sitting here with the CFO of UltraBotanica, Adam Payne. He's a dear friend of mine. And hey, Adam, what's up?
I think I'm actually the CEO, not the CFO.
CFO. Well, actually, you're trying to find a CFO. You've had to wear all those hats, right? For a long time.
CFO, CEO, COO.
Right.
You are our you're our CPE, which very few people really know what that is. It's a Chief Product Evangelist. And a unique name, which so fits kind of the your style and your role here at UltraBotanica. So, Josh, we met.
You and I normally get to do something which is so fun. Yeah. You have all these relationships with these awesome, innovative, functional, integrative, regenerative medicine specialists. We know compounding pharmacists. We know clinical nutritionists. And we normally get a chance to interview them. But today, I get a really cool opportunity.
Oh, you're going to interview me.
Yeah, see, I'm a guy that loves it. I'm a guy that loves the mystery. Right, I love.
Okay. So this is a surprise. That's why we're here today. I had no idea. So go for it, Josh.
I love a good mystery, and about 10 years ago, you and some colleagues at Oklahoma Medical Research Foundation and University of Oklahoma Health Sciences Center, you got approached by some people, and this is very much going to be a spoiler alert because. Mm-hmm. I love inventors too, by the way. I seem to attract them in my life, and introduce them to other inventors, so it's really fun. Mm. But anyway, you got approached with a conundrum, with a dilemma to try to solve with some colleagues, and the result of that, fast forward to today, has been hundreds of doctors worldwide, and literally now tens of thousands of people that have actually been helped with mobility issues with stiffness, issues with inflammation, and it all came down to a mystery that you guys were trying to solve out there. Remind me what you guys were up to 10 years ago when you got approached, Adam.
Well, it's actually almost 13 years ago at this point, Josh. Yeah. So, God takes us on a journey, and that journey really... we don't know the end as we're going on it, right? We just know that we're in the process. And I decided to, I had my conversion experience being a Jewish kid when I was about 20 years old. I met Jesus to the chagrin of my family. My mom's one piece of advice to me when I shared with her with all excitement my faith and this new experience I was having with the power of Christ inside of me, she was like, "Don't tell your grandfather. You'll give him a heart attack." And of course I tell him, and he was like, "Well, I don't understand any of that." Anyways, but I started my journey of surrender back, almost 40 years ago. Right. And the Lord took me on that journey. In 2008, I had a bad medical experience, and I had to resign I was CEO of a pharmaceutical company, and I had to resign from that because I couldn't medically keep going. And it brought me, Josh, to a different level of surrender. So at that point, I was part of a pharmaceutical company developing drugs in the inflammation space for inflammatory bowel disease and gastroenterology. I'd been working with a company in the rheumatology space, and we had been developing laboratory diagnostics to develop for rheumatoid arthritis that would measure the actual disease activity that was going on inside people with RA. And so God had prepared me through all of these life experiences with so much knowledge. And I thought, stupidly, because God has other plans sometimes. I thought in 2008, when I was at the pinnacle of my career, making close to $300,000 a year as the CEO of this pharmaceutical company, with bonuses still to come, I remember looking out my window and thinking, God, thank you for making this the purpose of my life, that I'm able to help people with these drugs.
What a setup that was, right?
Yeah.
That prayer.
And, fast forward 3 months later, and I'm in a hospital, and I had to resign, and all the things that happened with me, to me, through me, because of me, I found myself without a path. And I remember just surrendering again and saying, God, it doesn't matter anymore who I was, whether I was a CEO of a pharmaceutical company or not. I'm just going to trust you for whatever that path is. And out of the brokenness of my situation, I started working again, humbly, with a lab that was developing some interesting natural products. And Josh,
and of course, those are your favorite things to do.
No, they were not my favorite things to do. I grew up in a hippie home. My home, we had no furniture. We had beanbag chairs, and we had some crazy green vegetables that people were smoking in the basement.
You ran the complete opposite direction, didn't you?
Yeah, I was like the anti-hippie. And so when my dear friend, Mike Santola, he was a PhD, approached me and said, hey, why don't you come and help us? We're working on some natural products. And I was like, no, I don't want to do it. But God had some other ideas for me. And so I just said, you know what? I just need to breathe into this and learn and see what God has for me here. And Josh, that work started a journey. We ended up developing one of the only homeopathics that we've proven with clinical efficacy had a profound impact on eczema and psoriasis. And that technology was bought by Procter & Gamble. And it was just a phenomenal experience. But needless to say, Josh, in 2010, my company, because we started a new company, I just can't sit still, right? And the company was focused on these natural products. And so we were getting a reputation for this stuff.
And you lived in a hotbed of PhD researchers and MDs, right?
Yeah, no, this was the Presbyterian Research Parkway Foundation Park. And this was a business grouping, business offices / laboratories that was developed in order to help grow and build biotech companies in Oklahoma. So we had space there, and we were getting a reputation in natural products. So a dear friend of mine, a guy named B.G. Kurian, had heard about, actually he wasn't my friend at the time, but he just heard about all of our research on these natural products. And he came humbly up to my office one day and said, hey, Adam, I've heard you guys are working on some stuff in natural products. Excuse me. Could you maybe help us in what we're doing here? And I'm like, oh, sure. What are you guys cooking? And he was working in the labs of a guy named Hal Schofield. Hal Schofield is one of the the major researcher on the planet in Sjogren's syndrome, and one of the key researchers around the world in lupus. Two very debilitating autoimmune diseases.
Right.
Hal is just a wonderful guy, and BG's just a sweetheart of a man. And so when they approached me and said, hey, they're working on something, I was like, you know what? I don't know if I can be helpful here, but I can at least sit down with you guys and see what if I can be of any help to what you're doing. To my chagrin, and I know that's the second time I've used this word today, I learned that they were working on curcumin, which is the turmeric derivative. And for the second time in 3 years, I was like, no, not turmeric, not curcumin.
Right, because nobody had heard of curcumin, and nobody was talking about it in any of these poster presentations that you were attending, right?
No,
you never heard
it was actually just the opposite. Every single science conference I went to, there were at least a dozen or more posters about the wonders of curcumin on the inflammation cascade in the body. And I remember seeing all of these posters of this natural product in these autoimmune diseases and thinking to myself, wow, I guess there must be something there, but man, I'm glad I'm not working on that because I don't want to work on natural products.
Right.
I'm laughing because that's what my life is right now, right? Most of what we're doing now is working on curcumin.
Pretty much live, eat, breathe, sleep, right?
Yeah. It's ironic, but Josh, when I stuffed down my negative emotions and I really listened to what BG had going on, and they had the kernel of a good idea. So here's the problem with curcumin, and it's actually
and just for people to know, Adam, turmeric is a spice that I grew up having in my mom's. She'd get it out at Thanksgiving and sprinkle it on foods and stuff, and
it's
use
a
it at different
spice.
times.
It's
And
used for curry.
It's like a root, right? It's a rhizome.
Right, it's
But
a root. Yeah.
curcumin is just like a small little, maybe 2 or 3%, and that's kind of the magic bullet for this inflammation thing, right?
So we could go back in time a little bit. I'll go back in time, but then remind me what the key problem. I'll remind me to share about the
we'll
key
come
problem
back to the
here.
mystery. Absolutely.
So there are these class of researchers called epidemiologists. It's a fancy word for people that study the statistics of populations. And back in the 70s, global statistical data was just becoming available. Up until that time, people had collected global health data, but nobody had really collected it in such a way so that they could compare and contrast the health of multiple different populations on the face of the earth. What one of the key findings that they found, and this shocked the medical community back in the 70s, was that there were certain populations on the earth that had statistically significant lower rates of Alzheimer's, cancers, and inflammatory diseases. Now, Gesundheit. Now, what we didn't, there wasn't much diabetes at the time, but it turns out it was later in the analysis also diabetes. And so when they got this data, they're like, well, what is it? What differentiates? Why is this population down here in cancer and everywhere else on the face of the earth, it's way up here? I mean, it's like here or here. Mm-hmm. And when they peeled back the onion, it had nothing to do with genetics. So it had nothing to do with kind of the race of the people, had nothing to do with the climate, had nothing to do with the longitude that they were in. It had nothing to do with their education, the country, what they found was that it was in the food. And when they further peeled back the onion on the food, it had nothing to do with onions, by the way. When they further peeled back the information, they found that if turmeric was a daily part of their diet, then they had these lower rates. If it wasn't a daily part of their diet, it was up here. And so that sparked a whole bunch of research. That research ended up dissecting all of the different predominant molecules in the turmeric plant, and there's a lot of them. There's terpenes, there's different polyphenolic compounds, there's tumerones, different oils. When they finally pulled it apart, they found that there's 3 molecules in there that are based upon the curcumin molecule, and these are polyphenolic compounds. In other words, these are different structures, but they have phenolic rings inside of them that look like 2 little donut holes. And those are unique compounds that are in a class of bioactive plant compounds called polyphenols. And these polyphenolic compounds were the responsible molecules for this activity. When they tested these molecules in cancer cells, in inflammation models, it made the difference.
And so let's fast forward to this mystery, this problem that you needed to solve, because doing something in a petri dish, doing something on a cancer cell line study in vitro, what's the difference? What was the problem with human beings trying to consume this and get some kind of great result?
Now remember, the differences in the populations between here and here was if it was a daily part of the diet. The hope is that they were hoping that they could take the curcumin molecules, these polyphenolic compounds, and deliver them as a therapeutic agent into somebody's life so that they could kill the cancer cells or mitigate Alzheimer's or or inflammation diseases. And what they found was that the raw turmeric and or the extracted curcumin had little to no effect on the human body.
Ouch.
And the problem was they finally identified what the problem in a petri dish, in a test tube. It worked beautifully, but that was because they could dissolve those curcumin molecules into free bioavailable molecules that could interact with the cell. And
Right.
typically they would use DMSO or methyl alcohol or some other isopropyl alcohol to dissolve those curcumin molecules and get them to in a molecular state. What they learned what the problem was, Josh, is that these polyphenolic compounds, and this is the problem with all polyphenolic compounds, or
And there's
most of them.
dozens of them in
Hundreds
the natural
of
supplement space,
hundreds.
right? Yeah.
In the supplement space, there's dozens.
Yeah.
In nature, there's hundreds, if not thousands, of these molecules. Probably millions, if we really wanted to get down and identify them and measure them all. The problem is that these molecules bind to themselves. So you can imagine, here's the polyphenolic compound. What they like to do is bind to themselves. So when it's
So
bound
it
to
becomes
itself,
a cluster, right?
it
It's
becomes
just
a mess.
It's like rolling a snowball.
It's a hot mess.
It gets
It's-
bigger and bigger.
Yeah. And that's what you see is that these, when they extract the curcumin out of the turmeric root, you get these little crystals. And these crystals are insoluble, literally insoluble in water. It's not quite as bad, but it's close to like trying to dissolve quartz crystals and get silicone molecules out of it.
Almost like oil and water, right?
Yeah, that's a bad analogy. It just doesn't dissolve.
Yeah.
You had to take.
Good visual, bad chemistry there.
So they would pulverize the curcumin and give 8 to 12 grams of curcumin to people in cancer studies, which is a lot. That's like 16 capsules.
Gosh, that's like eating a meal.
Yeah, talk about
Right.
the cinnamon challenge. Tried doing the curcumin challenge, not pleasant. And they were only getting a 50 nanogram signature in the blood. That's 50 billionths of a gram curcumin was being seen in the blood after taking 8 to 12 grams of curcumin.
Which would mean no real result or effect in the individual for what they want to get out of it. Right?
And the results in the clinical studies were nil. Nothing. It wasn't of any benefit. So this is the problem. This on the background of all of this, BG came to me and asked, he said, I think I have a solution to the problem. And so why don't we go to break? And after the break, Josh, we will come back and talk about BG's solution to the problem.
So here we are, UltraLife Today, Adam Payne, CEO of UltraBotanica, and Josh Bellew, Chief Product Evangelist. We'll be right back.
Our mission is to take nature's most beloved botanicals and enhance them with our liquid protein scaffold technology. This helps it reach your cells faster and better. With exponentially enhanced bioavailability, you'll feel better every day. UltraBotanica, the feel-good curcumin.
So here we are again, UltraLife Today with CEO of UltraBotanica, Adam Payne. I'm Josh Bellew, Chief Product Evangelist. And we've been heading down this interesting road of how Adam and some colleagues discovered a way to get curcumin, which is a crystalline molecule that doesn't want to absorb in human beings. They can only seemingly do it in laboratory petri dishes. And Adam was presented with a problem from a colleague. And take us up right there with you and BG Kurian again, 12, 13 years ago, Adam.
So when BG approached me, I thought, oh, we figured this out. Well, I mean, somebody must have figured this out. Because there couldn't, it can't be that there's still a problem after decades of research. But BG approached me and said, look, Adam, there's still a problem with getting curcumin to absorb in the body. I'm like, okay, well, what's your solution, BG? And he said, well, my solution is just to boil curcumin in water and essentially to kinetically, by adding heat, to improve the soluble fraction of bioavailable curcumin molecules in a liquid matrix. In other words, he was making freely bioavailable curcumin molecules into dissolving it in the liquid. So rather than having to wait for the curcumin to dissolve if we take it in our bodies, he was pre-dissolving it into water,
Right.
and making that the dosage form. And to his chagrin, there we go. That's my word of the day.
I love it.
We made some of this stuff, and I sat around the table with some of my colleagues, and we took shots of it. And he was horrified. Literally, he was horrified.
Let's make a distinction between hypodermic shots and actually shot glasses.
Yeah, no, these were
right?
shot glasses.
Okay.
Yeah, these were shot glasses. These were not injections.
We don't want to have anybody get the idea they should boil this stuff up on the stove and inject it in their body.
No.
Right? Okay.
But he was horrified. He's like, "No, you did that? We need to do safety studies. We need to test this in animals first. We can't just take this into the body. We don't know what effect it will have." And
I like your style, brother. That's what I would have done.
Right? But I was like, BG, curcumin has been a part of everybody's diet for decades. There is no clinical study out there that has said that curcumin could in any way be toxic to the human body. And you know what happens? You know, the most surprising thing to me, and this is what really hooked me, Josh, we were sitting there, and literally in less than 5 minutes, we all felt this buzz.
Isn't it weird, Adam? Because it's something that almost just kind of seems to move from the neck up.
Yeah.
And you kind of look around, and it's like, did I just get
Am I imagining this?
Transported onto the Enterprise? What happened? Something
Yeah, no. I mean, you just, and I looked around at my colleagues. There were two of us sitting at the table, and I'm like, "Are you feeling this? Are you experiencing?" And they're like, "Yeah." And so what hit me, Josh, more than anything, one, I was like, "Wow, hey, it's kind of a nice feeling." It wasn't a bad feeling.
Here, and there's something here, right?
But what hit me, Josh, was two things. One, it's getting in the body, and two, it's getting in my brain.
Wow.
And
they have a hard time finding drugs that do that. Adam.
Yeah.
Wow.
Well, I mean, this was astonishing to me. And so I went from being kind of like, yeah, I'll help you, BG, whatever ideas you have, to like, there's something here. Now the problem with liquid curcumin is that it wasn't as stable as we wanted to. You'd have to take home a bottle of stuff for one dose. Not to you're not going to carry a crate of curcumin bottles from the store.
You have to figure out a way to preserve it.
Yeah.
You have to figure out a way to keep it from staining your lip yellow and your tongue.
That's true. Yeah, it stained horribly. It's still not a bad way to get curcumin into the body, but it was too bulky and it wasn't that stable.
Yeah,
and there wasn't really an efficient way of doing it to get it into that form. But that started in 2010. That started an intense research program at my company, and I had like 5 different technologies that I was developing as CEO of my previous company. And a couple of insights later, it turned into our UltraCur technology, which is, we call it LPS technology, which stands for liquid protein scaffolding. So it started with the liquid, and later we added this thing called protein scaffolding. And that was the result of some research that myself and a guy named John Chansey did back in 2012 and 2013. So let me tell the story about how this happened. So we really weren't doing anything. So scroll back to 2012. We really weren't doing anything serious with the curcumin technology. BG and I tried to get some grants, and we thought about ways to potentially commercialize the technology, but really it wasn't very efficient, and we were still so early in the development pathway. But in 2012, BG came to me and said this. He said, "Adam, I've been using liquid curcumin to stain proteins and what's called a page gel. And this is a scientific method that's used in order to break apart proteins and to look at all of the different molecules that are in a complex protein environment. So you take a drop of whatever gamish of proteins that you want to study, you put it on one edge of the gel, and you apply an electric current, and that electric current separates the proteins into its different molecular weights. The problem is you can't see the proteins unless you stain them. And what BG was doing was staining the proteins with the liquid curcumin. And he was doing it before the proteins separated. And so this blew my mind. It really did. Most people would think, oh, whatever. What does that mean? And why do I really care? What I learned from what he was doing was this, two things, Josh. The first is that curcumin would bind to protein before it would bind to itself. That means that, because normally if you take soluble curcumin and drop it in water, or in a non-water alkaline environment or acidic environment, it precipitates. It starts binding to itself and coming
just
out of
wants
solution.
to find all its cousins and brothers and sisters
and have a big hug.
Have a party.
Yep. But in this case, it wasn't doing that. It was actually binding to the proteins before it bound to itself. And so I thought to myself, well, if it's binding to the proteins before it's binding to itself, maybe we can use protein as a scaffold on which we can deliver the curcumin molecule. So that thought came to me as follows. John and I were sitting in my office, and he had just come back. He's a large animal horse vet. He works at the racetrack here in Oklahoma. He is responsible for hundreds of horses on a daily basis. And he came back into my office just in a foul mood. He had been testing an ethanolic tincture of curcumin on horses because we can get a lot more curcumin dissolved into ethanol.
And for those that don't know what ethanol is, back in my junior high days, they called stuff Everclear, didn't they?
It's alcohol.
Not like it, right?
It's drinkable alcohol, right?
Right.
It's not the stuff that'll make you blind,
Right,
unless you drink
exactly.
way too much of it, right? So he had just dosed a horse with an ethanol tincture of curcumin, and he was like, "Man, we got to think of something different." I got that horse drunk, and a drunk horse is not a happy horse.
Yeah, not $1 million racehorse, right?
No, but we had good. We had what do you call it? We had good intentions. We were trying to help the horses because they get horrible inflammation after they run. It's just really hard on their joints. And so John and I were sitting in my office, and I was dropping some of this tincture of curcumin onto my finger with a dropper. Where's my finger there? And I was dropping it on there. And the curcumin was actually going into the structure of my skin, and it would stain the structure of my skin so that even if I washed my finger, it wouldn't come off for like 3 or 4 days because it was inside the structure of my skin on the proteins. Remember, curcumin would bind to the proteins before it would bind to itself. And so I was sitting there dropping this curcumin tincture on my finger. I thought, well, why don't we just use protein as a way to carry these molecules of curcumin in the body? Because they will evenly distribute themselves on the protein like a Christmas tree. And then maybe as the body's digesting the protein, those molecules will become bioactive in the body. And so, Josh, this is exactly what we tried. And the results were off the chart. So why don't we go to break? And after I come back from break, I will share some of the results that we got from this new invention that we had come up with.
Once again, you've joined UltraLife Today.
Our mission is to take nature's most beloved botanicals and enhance them with our liquid protein scaffold technology. This helps it reach your cells faster and better. With exponentially enhanced bioavailability, you'll feel better every day. UltraBotanica, the feel-good curcumin.